Welcome back to Concepts of Biology. We'll continue to read Chapter 3, Section 3.3, Eukaryotic Cells, the Cytoskeleton. By the end of this video, you will be able to state the role of the cytoskeleton. If you were to remove all the organelles from a cell, would the plasma membrane and the cytoplasm be the only components left? No. Within the cytoplasm, there would still be ions and organic molecules, plus a network of protein fibers that helps to maintain the shape of the cell, secures certain organelles in specific positions, allows cytoplasm and vesicles to move within the cell, and enables unicellular organisms to move independently. Collectively, this network of protein fibers is known as the cytoskeleton. There are three types of fibers within the cytoskeleton: microfilaments, also known as actin filaments, intermediate filaments, and microtubules. Figure 3.9. Microfilaments, intermediate filaments, and microtubules compose a cell's cytoskeleton. Microfilaments are the thinnest of the cytoskeletal fibers and function in moving cellular components, for example, during cell division. They also maintain the structure of microvilli, the extensive folding of the plasma membrane found in cells dedicated to absorption. These components are also common in muscle cells and are responsible for muscle cell contraction. Intermediate filaments are of intermediate diameter and have structural functions such as maintaining the shape of the cell and anchoring organelles. Keratin, the compound that strengthens hair and nails, forms one type of intermediate filament. Microtubules are the thickest of the cytoskeletal fibers. These are hollow tubes that can dissolve and reform quickly. Microtubules guide organelle movement and are the structures that pull chromosomes to their poles during cell division. They are also the structural components of flagella and cilia. In cilia and flagella, the microtubules are organized as a circle of 9 double microtubules on the outside and 2 microtubules in the center. The centrosome is a region near the nucleus of animal cells that functions as a microtubule organizing center. It contains a pair of centrioles, 2 structures that lie perpendicular to each other. Each centriole is a cylinder of 9 triplets of microtubules. The centrosome replicates itself before a cell divides, and the centrioles play a role in pulling the duplicated chromosomes to opposite ends of the dividing cell. However, the exact function of the centrioles in cell division is not clear, since cells that have the centrioles removed can still divide, and plant cells which lack centrioles are capable of cell division. Flagella and cilia. Flagella, or singular flagellum, are long, hair-like structures that extend from the plasma membrane and are used to move an entire cell, for example sperm, euglena. When present, the cell has just one flagellum or a few flagella. When cilia, singular cilium, are present, however, they are many in number and extend along the entire surface of the plasma membrane. They are short, hair-like structures that are used to move entire cells, such as paramecium, or move substances along the outer surface of the cell, for example, the cilia of cells lining the fallopian tubes that move the ovum toward the uterus, or cilia lining the cells of the respiratory tract that move particulate matter toward the throat that mucus has trapped. This ends Chapter 3, Section 3.3. Eukaryotic cells, the cytoskeleton. In our next video, we'll read Section 3.3, the endomembrane system.